Hi class, this is your instructor, Skylar Huff, and this is gene regulation. So this chapter begins information for your next test, and the next test will include chapters 14, 15, and 16. Let us now begin. Each type of cell in a multicellular organism has a characteristic shape, carries out very specific activities, and makes a distinct set of proteins. With few exceptions, however, all cells in an organism contain the same genetic information. Why, then, aren't cells identical in structure, molecular composition, and function? Well, it's because cells differ, of course, in gene expression, and it's regulated. So only certain subsets of the total genetic information are expressed in any given cell. What mechanisms control gene expression? Well, they consider, of course, gene coding for a protein that is an enzyme. Expressing that gene involves three basic steps: transcribing the gene from messenger RNA, translating the gene from mRNA to protein, and activating the protein so it can catalyze a specific reaction in the cell. Thus, gene expression results from a series of processes, each of which regulated in many ways. The control of mechanisms use various signals, some originating within the cell, and others coming from other cells or from the environment. These signals interact with the DNA, RNA, or protein. Mechanisms that regulate gene expression include control of the amount of mRNA transcribed, of the rate of translation of mRNA, and of the activity of the protein. These controls are accomplished in several ways. For example, the rate of transcription and the rate of mRNA degradation both control the amount of available mRNA. So the mechanisms here aren't just unique to, of course, one type of organism or another. Although bacteria are not multicellular, regulation of gene expression is also essential for their survival. Gene regulation in bacteria often involves controlling the transcription of genes whose products are involved in resource use. In eukaryotes, by contrast, fine-tuning of control systems occurs at all levels of gene regulation. And that involves all level of gene regulation and is consistent with the greater complexity of eukaryotic cells and the need for developmental controls in multicellular organisms. So keep all of this in mind as we continue through this brief chapter, and this chapter will not be long at all, class. So let us begin. So the main requirement of bacterial gene regulation is the production of enzymes and other proteins when needed. So bacterial and eukaryotic cells, they use distinctly different mechanisms of gene regulation and is based on a specific set of requirements of the organism. So bacterial cells, they can exist independently, and each cell performs all of its own essential functions. Because they grow rapidly and have relatively short time frames between cell division events, bacteria carry fewer chemical components than eukaryotic cells. So expression of bacterial genes occurs when it helps bacteria adjust to their environment, including any food that might be present in the surroundings. The primary requirement of bacterial gene regulation is the production of enzymes and other proteins when needed, and transcriptional level control is the most efficient mechanism. So, with that, it's the organization of related genes into groups that are rapidly turned on and off as units that allows the synthesis of only gene products needed at a particular time. This type of regulation requires rapid turnover of mRNA molecules, and by doing that, it prevents those messages from accumulating and continuing to be translated when they're not needed. Bacteria regulate rarely regulate enzyme levels by degrading proteins. Once the synthesis of a protein ends, the previously synthesized protein molecules are diluted so rapidly in subsequent cell divisions that breaking them down is usually not necessary. Only when cells are starved or deprived of essential amino acids do protein digesting enzymes recycle amino acids by breaking down the proteins no longer needed for survival. So eukaryotic cells have different regulatory requirements. Although transcriptional level control predominates, control at other levels of gene expression is also very important, especially in multicellular organisms in which groups of cells cooperate with one another in a division of labor. And it's because a single gene is regulated in different ways and in different types of cells, the eukaryotic gene regulation is complex. Eukaryotic cells usually have a long lifespan during which they need to maintain homeostasis while responding to different stimuli. New enzymes are not necessarily synthesized each time the cell responds to a stimulus. Instead, in many instances, preformed enzymes and other proteins are rapidly converted from an inactive to an active state. Some cells have a large store of inactive RNA. For example, the mRNA of an egg becomes activated when the egg is fertilized. So much gene regulation in multicellular organisms is focused on the differential expression of genes in the cells in various tissues. Each type of cell has certain genes that are active and other genes that may never be used. For example, developing red blood cells produce the oxygen transport protein hemoglobin. Muscle cells, on the other hand, never produce hemoglobin, but instead produce myoglobin, a related protein coded for by a different gene that stores oxygen in muscle tissue. Apparently, the selective advantages of cell cooperation in multicellular eukaryotes far outweigh the detrimental effects of carrying a load of inactive genes through many cell divisions. The domesticated pig provides another example of how gene regulation in multicellular organisms affects expression in different tissues. A genetic mutation in pigs makes them develop more muscle tissue. The mutation is in a gene designated insulin-like growth factor 2, or IGF-2. This, of course, codes for a protein produced by both muscle and liver tissues. Biologists have found a base substitution mutation in the IGF-2 gene makes the gene 3 times more active in pig muscles, resulting in leaner meat. However, the mutation does not change expression of the gene in liver cells. Interestingly, this mutation is not in the protein coding protein of the gene. It is not in the protein coding portion of the gene. It is in the regulatory mutation located in an intron. So it's pretty amazing that you find this class. Instead, it's regulatory mutation located in an intron. And this goes back to where you all learned about post-transcriptional modification with introns and exons. So we'll now get directly to gene regulation in bacteria to finish things up. So in bacteria, for example, E. coli has been used, Escherichia coli, the common bacterium found in the intestines of humans and other mammals. It has 4,288 genes that code for proteins, and approximately 60% of which have known functions. Some of these genes code for proteins that are always needed, such as enzymes involved in glycolysis. These genes, which are constantly transcribed, are called constitutive genes. So these constitutive genes, and as biologists have described them, they constitutively expressed. Other proteins are needed only when the bacterium is growing under certain conditions. For instance, bacteria living in the colon of an adult cow, not normally exposed to the milk sugar lactose, which of course is a disaccharide. If those cells had ended up in the colon of a calf, however, they would have lactose available as a source of energy. However, they would have lactose available there. So this situation presents a dilemma. Should a bacterial cell invest energy in materials to produce lactose metabolizing enzymes just in case it ends up in the digestive system of a calf? So if E. coli cells could not produce those enzymes, however, they might starve in the middle of an abundant food supply. Thus, E. coli functions by regulating many enzymes to use available organic molecules efficiently. Cell metabolic activity is controlled in two ways. One is by regulating the activity of certain enzymes, and that's just how effectively an enzyme molecule works, and the other is by regulating the number of enzyme molecules present in the cell. So some enzymes are regulated in both ways. So let's get down to what is known as the operon. Operons in bacteria. So this comes from the work of Jacob and Menard from the 1960s, particularly 1961. So what happened here is these French researchers were credited with the first demonstration of gene regulation at the biochemical level through their elegant studies on the genes that code for the enzymes that metabolize lactose. In 1965, they received the Nobel Prize in Physiology or Medicine for their discoveries relating to genetic control of enzymes. To use lactose as an energy source, E. coli first cleave the sugar into the monosaccharide glucose and galactose. Those are what make up the disaccharide, GLAS being lactose. And they do that using the enzyme beta-galactosidase. Another enzyme converts galactose to glucose, and the enzymes in the glycolysis pathway further break down the resulting two glucose molecules. This is all review. So to ensure you understand what's happening here, an operon is a gene complex consisting of a group of structural genes with related functions, plus the closely linked DNA sequences responsible for controlling them. So E. coli cells growing on glucose produce very little beta-galactosidase enzyme. However, each cell grown on lactose as the sole carbon source has several thousand beta-galactosidase molecules, accounting for about 3% of the cell's total protein. So amounts of the two other enzymes, lactose permease and galactosidase transacetyl also increase when the cells grow on lactose. So the cell needs permease to transport lactose efficiently across the plasma membrane, and without it, only small amounts of lactose enter the cell. Transacetylase may function in a minor aspect of lactose metabolism, although its role is not yet clear. So what Jacob and Menard identified were mutant strains of E. coli in which a single genetic defect wiped out all three enzymes. This finding, along with other information, led the researchers to conclude that the DNA coding sequences for all three enzymes are linked as a unit on the bacterial DNA and are controlled by a common mechanism. So with that, each enzyme coding sequence is a structural gene. Hence, they coined the term Operon. The structural genes of the Operon, or lactose operon, as we'll get to now, are LAC-Z, LAC-Y, and LAC-A, which of course code for beta-galactosidase, lactose permease, and transacetylase, respectively. So the transcription of the lac operon begins with, of course, RNA polymerase, which binds to a single promoter region upstream from the coding sequences. It then proceeds to transcribe the DNA, forming that single RNA molecule that contains the coding information for all three enzymes. Each enzyme coding sequence on this mRNA contains its own start and stop codons. Thus, the mRNA is translated to form three separate polypeptides. Because all three enzymes are translated from the same mRNA molecule, their synthesis is coordinated by turning a single molecular switch on or off, which of course is that operator. The switch that controls mRNA synthesis, as I mentioned, class, is the operator, which is a sequence of bases upstream from the first structural gene in the operon. In the absence of lactose, a repressor protein, get it? Repressor. So a repressor protein called lactose repressor binds tightly to the operator. So RNA polymerase binds to the promoter, but is blocked from transcribing the protein coding genes for the lac operon. The lactose repressor protein is enclosed by a repressor gene, which in this case is a protein coding gene located upstream from the promoter site. So unlike the lac operon genes, the repressor gene is constitutively expressed and therefore constantly transcribed. So the cell continuously produces a small amount of that repressor protein. The repressor protein binds specifically to the lac operator sequence, and when cells grow in the absence of lactose, a repressor molecule nearly always occupies the operator site. When the operator site is briefly free of that repressor, the cell synthesizes a small amount of mRNA. However, the cell synthesizes very few enzyme molecules because E. coli mRNA is degraded rapidly. The half-life is about 2 to 4 minutes, class. So to ensure that you understand this, class, lactose turns on or induces the transcription of the lac operon because the lactose repressor protein contains a second functional region known as an allosteric site that is separate from its DNA binding site. This allosteric site binds to the allolactose, a structural isomer made from lactose. And if lactose is in the growth medium, a few molecules enter the cell and are converted to allolactose by the few beta-galactosidase molecules present. So the binding of a molecule of allolactose to the repressor alters the shape of the protein, so its DNA binding site is no longer recognized as the operator. When the repressor molecules have allolactose bound to them and are therefore inactivated, RNA polymerase can actively transcribe the structural genes of the operon. So the E. coli cells continue to produce beta-galactosidase and the other lac operon enzymes until it uses up virtually all lactose. When the intracellular level of lactose drops, so too does the concentration of allolactose. Therefore, allolactose dissociates from the repressor protein, which then assumes the shape, and the repressor binds to the operator region and shuts down the transcription of the operon. So here we go. I'll begin to explain what I just did with you all. So here, class, upstream is that repressor gene. That repressor class, once, of course, it is bound to the operator, that on-off switch, it blocks the access of RNA polymerase to the DNA strand. Without RNA polymerase, class, it cannot transcribe those genes to break down any lactose. And of course, in this case, lactose is not present because the repressor is there in its active form, bound to the operator. So I hope that you all can understand what I'm showing you here. And if you cannot, please let me know. And this is the key point, class, you'll find on page 300 in figure 14.2. Figure 14.2. So here we have the repressor gene yet and still upstream. However, what has happened now is we have our inducer, and in this case, the inducer is allolactose. And allolactose is here, if you all can remember back from what I just mentioned, is that allolactose is a structural isomer made from lactose. So, since the inducer is here, it now, class, has inactivated. It now, class, has inactivated, which was once that repressor protein, which then, class, allows RNA polymerase to access the strand of DNA to transcribe, class, this lac Z called beta-galactosidase, to transcribe lactose permease, and to transcribe, class, transacetylase. And these three enzymes are the enzymes needed for the metabolism of lactose. So that is the Operon system, class, and DNA control mechanisms in a prokaryote. So, as the lac Operon has been studied and was studied, it's just curious, of course, maybe how do they elucidate the function of lac Operon? Well, their approach involved using mutant strains. And even today, class, researchers use mutant strains. So the genetic crosses of mutant strains allowed the investigators to determine the map of positions in a linear order of the genes on the DNA to infer normal gene functions. So by studying what happens when they are missing or altered, the researchers usually combine this information with the results of direct biochemical studies. So if you look closely here, it just shows you that the mutation affects on one or all the enzymes involved. And as you're seeing this diagram is exactly how Jacob and Bernard inferred their conclusions based on their studies. So if they were to think that, yes, it's just one enzyme affected, then of course the mutation isn't a structural gene. However, if they were to affect class all three enzymes, then of course the mutation affects gene regulation. So it took time, and it was a process, but it was definitely done, class, and done quite well. So now I'll get briefly here, class, to inducible genes. So an inducible gene is not transcribed unless the inducer activates its repressor. So in our case, class, what we just learned about, of course, was allolactase, a definite inducer. A definite inducer. And of course, that would mean that the lac Operon, class, is an inducible operon. Or in other words, a repressor usually controls the inducible gene or operon by keeping it turned off. Repressible genes. So as I get down to repressible genes, so these genes are usually turned on and they are turned off only under certain conditions. And in most cases, the molecular signal for regulating these genes is the end product of the metabolic pathway. That would be kind of like we learned about when we got to what is known as feedback, and then, of course, feedback inhibition. So, having put it that way, the molecular signal for regulating these genes is usually that end product, usually that end product. And this kind of gets us to a different example called the trip operon, which is, of course, here for TRP. Alrighty then. So the trip operon, and this is, it's stated that because this is the tryptophan operon, and it's an example of a repressible system. So in both E. coli and related bacterium, and a related bacterium known as Salmonella, the trip operon consists of 5 structural genes that code for the enzymes needed to synthesize the amino acid tryptophan. These genes are clustered as a transcriptional unit with a single promoter and a single operator. A distant repressor gene codes for diffusible repressor protein, which differs from the lactose repressor in the cell that synthesizes it in an inactive form that cannot bind to the operator region of the trip operon. The DNA binding site of the repressor becomes effective or active only when tryptophan, its co-repressor, binds to an allosteric site on the repressor. So when intracellular tryptophan levels are low, the repressor protein is inactive and cannot bind to the operator region of the DNA. So it's the enzymes that are required for tryptophan synthesis are produced, and the intracellular concentration of tryptophan increases. So some of it binds to the repressor, altering the repressor shape, so it binds tightly to the operator, which switches the operon off, thereby blocking transcription. So briefly here, class, this is the trip operon. So if you look closely, here is that repressor gene. So the repressor gene class, as just stated in this, and of course this is our example class about those being repressible operons, the repressor here class is inactivated. Since it's not activated, RNA polymerase can access the strand of DNA to transcribe and then of course allow for the transcription of the protein. Well, transcribe is the same thing, but the translation of these differing proteins here, these differing genes, which you see there, class, transcribed and translated. So these are the enzymes of the tryptophan biosynthetic pathway. However, here in this case, in the presence of tryptophan, yes, we're still here with that repressor gene, which is, of course, yes, it's inactivated. Now that we have our co-repressor here, our co-repressor will bind to the inactivated repressor, activating the repressor to bind to that operator. So this is a repressible operon, and this example class is a trip operon as that example. So of course, negative regulators and positive regulators. I won't spend much time, class, going over the negative and positive regulation, but I do want to say that the features of both of these have been described as negative control, where it's a mechanism that is a regulatory, in which that the DNA binding regulatory protein is a repressor that turns off the transcription of a gene. So in positive control, that regulation activates proteins that bind to the DNA and of course simulate the transcription of the gene. So the lac operon is controlled by both negative and positive activated proteins. And I won't go through the process of this catabolic activated protein. I will not go through that process. And of course, this gives us a great overview of the process and how it works. All right, so transcription of constitutive genes. So you all have gotten a bit of information about such. But the genes coding for repressor or activated proteins, they regulate those metabolic enzymes and are usually constitutive and produce their proteins constantly, which you all knew about. So we get here, class, back to feedback inhibition by getting, of course, after a certain level of that enzyme has been produced, that level, class, will feed back to, of course, shut off the production of more and more of that enzyme. And finally, we've now made to 14.3, the last section, which is gene regulation in eukaryotic cells. So eukaryotes have transcriptional, post-transcriptional, translational, and post-translational gene control mechanisms. So eukaryotic genes are not typically arranged in operon-like clusters, and each eukaryotic gene has specific regulatory sequences that control transcription. So, of course, that means that some genes are constitutive and others are inducible. So some inducible genes respond to environmental threats such as heavy metal ingestion, viral infection, or even heat shock, such as the heat shock protein. Some genes are inducible only during certain periods of the lifespan. And then some genes are under tissue-specific regulation. So transcription requires a transcription initiation site plus a promoter. So certain promoter elements have regulatory functions that facilitate the expression of the gene, and the mutation in these elements reduce the rate of transmission, such as that top box, which I didn't mention much of. So enhancers and silencers regulate the gene on the same DNA molecule from very long distances. So of course the enhancer, they help form that active transcription initiation complex, and the silencers are those that decrease transcription. So as you all can see, here is the top box, part of the promoter region, and of course the gene downstream from it. So of course upstream from each of which class we have them both being of course the enhancer and the silencer. This is what I was referring to class shortly ago. So the transcription factors are of course here, and these are what help you prepare for transcription. And these are more or less class, just DNA binding proteins that regulate transcription in eukaryotes. So transcription factors, which are here class, generally they're bound to the top box of the promoter region, and they are required for RNA polymerase to bind to the DNA. So there we have it. We have the top box, an enhancer way upstream, and ultimately class there are transcription factors that are there for that transcription initiation complex. So chromosome organization also has an effect on the expression of some genes. I won't spend a lot of time with how chromosomes are organized, but of course active genes are associated more with a more loosely packed chromatin structure called euchromatin. So the exposure of DNA in euchromatin lets it interact with transcription factors and other regulatory proteins. So there we have it. So once a gene has been turned off by some other means, what happens is it's a process class in eukaryotes. And what I'm getting at is it's called DNA methylation. By adding a heavy coat of methyl groups, it prevents transcription. I can't say it any easier than that. And even to contrast that, it's called histone acetylation. So I showed histones here. So those smaller portions class known as histones, well, it's called histone acetylation. So by attaching acetyl groups, they allow RNA polymerase and transcription factors, hence allowing transcription to occur. That's by acetylation, which contrasts methylation, the adding of those heavy methyl groups. So with that class, this has been Chapter 14. Farewell class. The chapter is quite short. Make sure you all know about methylation and acetylation, not to mention class what are known as the Operon system for your test.